Thank you very much for the kind invitation to present our data regarding the targeting of cellular and subcellular mechanisms to treat heart failure. As you know, heart failure is becoming an increasing problem because of the higher incidence, and it comes in different flavors, meaning that there's heart failure with reduced ejection fraction, which can be due to ischemia or hypertrophy, or heart failure with preserved ejection fraction, which has many different origins and different features. Understanding the pathophysiology of these different types of heart failures and maybe even subclassifying heart failure subtypes may be very important in order to target treatments in the future. It may be also important to note that the different types of heart failure have different microenvironments and different changes in the cells in the context of cardiomyocyte communication, which may be important to keep into mind if you consider novel therapeutic developments. To get some insights into human heart failure subtypes, we used the novel technologies which became popular in the last years, namely single nuclei sequencing, where you can define the transcriptome of each cell in a tissue, which allows you to define the transcriptome in each cell type instead of having a bulk mixture of RNA transcripts which cover all different cell types before. In contrast to a smoothie-like impression of this bulk RNA approach, the single cell sequencing gives you really flavors of each individual cell, as also highlighted it in this cartoon, which is brought by my student. Using this technology, we then investigated the transcriptome of, first of all, patients with aortic stenosis. These are patients with hypertrophy, and merged these data sets with human heart tissue, which is available and published in a bioinformatic approach called Harmony. By pooling these data, we were able now to address the transcriptomic signatures in cardiomyocytes, fibroblasts, endothelial cells, and other cell types in the heart of these patient samples. And of course, this is just the beginning. We have now built up larger data sets, which also have breadth and have breadth to contribute to these data. But starting with the question, how is hypertrophy affecting the cardiac environment? We use the single cell sequencing data with a particular interest in also cells around cardiomyocytes. So you know cardiomyocytes are the powerhouse of the heart, of course, but also other cell types such as fibroblasts, the vasculature, and also a balanced immune response is very important to keep and maintain a healthy heart. And the other way around, this can all contribute to cardiac dysfunction. To address the interaction of these cell types, we first of all use the bioinformatic approach. This is based on the understanding that cell A, which is expressing a ligand, can be a membrane-formed ligand or a soluble ligand, and cell B, which is expressing the receptor, could in principle interact. While cell C, which has no ligand or no receptor, can of course not interact with cell type A or B. Using such an approach and the potential ligand-receptor interaction tool and a weighted analysis, we then compared healthy hearts and hypertrophied hearts. All these lines here showed a potential communication between cardiomyocytes. This is now the lines in red, and different other cell types of the heart, for example, endothelial cells and fibroblasts. And you can easily appreciate that given by these red arrows here, that these lines, if they are thicker, they depict more signals and more intense signals, that there is really a lot of communication ongoing. If you see the same picture in the hypertrophied hearts, these lines here become thinner, indicating that the number and rate, so interaction power between cardiomyocytes and other cell types, particularly to endothelial cells, becomes less. To study what this means and quantify these intensity strengths, this is now the incoming signals of cardiomyocytes, either in healthy hearts or in patients with aortic stenosis hearts. And you can appreciate that in healthy hearts, the interaction is much higher, for example, to endothelial cells as compared to the diseased hearts. And this is true for more or less all cell types in the heart, suggesting that there is a reduction in incoming signals to cardiomyocytes in these disease contexts. So being a vascular biology lab, of course, we were particularly interested in these findings showing that endothelial interactions are declined in disease context. And as you can see here, we now dig deeper into what signals are changed in interactions. And these are now the downregulated signals in aortic stenosis patients. You see here again the communication between cardiomyocytes and endothelial cells. And among the top candidates were many ephrin and ephrin receptor interactions, which were disturbed and downregulated in aortic stenosis patients. In addition, and also I will not talk about it, there are also VEGF, which is a very powerful vascular growth factor family members, which also showed a decline in interaction. But coming back to ephrin receptor interactions, we were particularly interested in these interactions because it had not been well studied in this disease context, although ephrins and ephrin receptors are very well known to be important for development, tissue fibrosis, inflammation, and angiogenesis. This is now a scheme showing you how ephrins and ephrin receptors interact. So the ephrins are usually, as I said, working as agonists, and they can activate ephrin receptors, but there is also a reverse signaling. So this really leads to an interaction between cells. There are many ephrin receptors and ephrin ligands, and you can see here the two I marked in red here, which are highly expressed in cardiomyocytes and which can potentially interact with the Ephrin B2, which is the agonist, which was also highly expressed. This shows you the data. So these are the Ephrin receptors in cardiomyocytes. As I said, Ephrin B1 and Ephrin A4 are the highest expressed ones. And you easily can appreciate that in healthy hearts, there's a lot of expression of Ephrin B1 in healthy cardiomyocytes. And this is almost close to absence in the diseased hearts. So each of these lines represent the intensity of expression in each cell. And you can really see only very few cells have still some expression in the diseased heart. And the downregulation was most profound in the Ephrin B1 receptor because this was leading us to the further investigation of this Ephrin B1 receptor function in cardiomyocytes. Some more details regarding the data. So with bioinformatic predictions, you would need to want to know how this really looks in the raw data. These are now Ephrin B1 receptor expression in each cell. You see here the dots representing cells, and you see here in the healthy heart many more Ephrin B1 highly expressing cells compared to the diseased hearts. And you can even see if you look in the individual samples, so these are 14 healthy controls and the 5 patients, there's really a clear distribution of highly expressing cells in the healthy hearts, while this is really absent in the diseased heart. And we also confirmed this on protein level because these are all RNA expression data and could show that indeed also protein expression of Ephrin B1 is reduced in the hypertrophied heart in collaboration with Benjamin Neder and his group in Heidelberg. You can see here the quantification below. Of course, the key question was, what is this meaning for the cardiomyocyte function and for the cellular communication? As said, Ephrin B1 is a membrane-bound receptor, and you see here in these cartoons and these UMAP plots that cardiomyocytes express very high levels of the Ephrin B1 receptor. And we were looking for ligands, and as said, there's a prediction that Ephrin B2 could be the ligand for Ephrin B1, and this is interestingly very highly expressed in endothelial cells and almost absent in the other clusters, suggesting that maybe endothelial cell Ephrin B2 could communicate to cardiomyocytes via the Ephrin B1 receptor, and this might keep cardiomyocytes healthy. To see whether this is really working as we predicted, we first of all tested the effects of recombinant Ephrin B2 and showed that this is indeed inducing the phosphorylation of the Ephrin B1 receptor, as you can see here in these Western blot analysis. With this in mind, we then questioned how these Ephrin B2, Ephrin B1 crosstalk may be contributing to healthy cardiomyocytes. And to get some first insights on the effect of Ephrin B1 receptor stimulation, we used recombinant Ephrin B2 and added this to classical hypertrophy assays. So you see here the induction of hypertrophy in cardiomyocytes by phenylephrine. And then we added recombinant Ephrin B2 to check these effects and could appreciate here that there is a reduction in hypertrophy induction induced by phenylephrine under these conditions. You can also see this here in these representative images. Also, we use the more physiological organoid model. This is a human cardiac organoid model in collaboration with Chaya Krishnan. You can again see here this hypertrophy response in response to phenylephrine. And as quantified below, again, if we add recombinant Ephrin-B2, this hypertrophy was reduced also in this more physiological setup, suggesting that there is an induction, a prevention of induction of hypertrophy by recombinant Ephrin-B2 ligand. So, of course, recombinant Ephrin-B2 may not do the same as endothelial cell Ephrin-B2. So we really studied how endothelial cells could communicate to cardiomyocytes and did so by silencing Ephrin-B2 in endothelial cells and then co-cultured these endothelial cells with cardiomyocytes. And then on the left-hand side, you see most impressive results. So this is now the co-culture. Here you can see the cardiomyocytes, and the bottom you see the endothelial cells, and you can appreciate, and these are the control conditions, and these are the conditions where we depleted Ephrin-B2 in endothelial cells. And you can appreciate the cardiomyocytes look bigger. So the removal of Ephrin-B2 in endothelial cells leads to an induction of hypertrophic responses in cardiomyocytes. This is quantified here on the right-hand side as that silencing of Ephrin-B2 induced cardiomyocyte hypertrophy. We could also see a decline in cardiomyocyte contractility and an induction of cardiac stress genes, so NPPA, NPPB, or ACTA1, all were significantly upregulated if we silence Ephrin-B2 in endothelial cells. So this means that the lack of Ephrin-B2 in endothelial cells is sufficient to induce cardiomyocyte stress responses. So if this is the case, we of course question whether we can also protect the cardiomyocytes by augmenting Ephrin-B1 receptor expression. And we did so by overexpressing Ephrin-B1 by an AAV approach. And as done before, we stimulated cardiomyocyte hypertrophy by phenylephrine induction, and then used the Ephrin-B1 AAV. And indeed, you can see here that there is really a protective response or a decline in phenylephrine response, as shown here. These data suggest that we have a striking change in cell communication in the diseased hearts, particularly the crosstalk of cardiomyocytes is changed, and there is also an activation of a lack of activation and communication between endothelial cells and cardiomyocytes, which leads to potentially augmented hypertrophy response, and this is mediated by Ephrin-B1 receptor expression. And then my students concluded that, of course, this is all about cell communication, and in this cartoon you can depict what this may mean for your heart. So usually under healthy conditions, the endothelial cell is continuously communicating with the cardiomyocytes by the Ephrin-B2 interaction with the receptor, while in the diseased heart there is no longer expression of the receptor, and of course this prevents the activation of endothelial cell cardiomyocyte responses, and this may contribute to hypertrophy responses. With this, I would like to conclude, and of course, we would imply that targeting cell communication pathways may be one aspect of a new therapeutic option. Of course, this needs to be further developed. It's early times, but of course, this could be an option in addition to targeting fibrosis, to vascular therapies, to immune therapies, or matrix therapies, or even senescence therapies. All of these have valid implications, but maybe targeting cell communication in this context may be considered as well, maybe also in combination with these approaches. With this, I would like to thank the people who did the study, particularly our collaborators, Benjamin Meder, I already mentioned, and the other partners, and would like to thank you for your attention.